Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speakers. So thank you for joining us today. It is now my pleasure to introduce our speakers, Andeka Ocio from Progenika, a Grifols company, and Rob Martindale from Masterflex.
Thank you, Leah. I am Andeka Ocio, Manufacturing and Process Validation Technician at Grifols, and I am glad to share this webinar with Rob, which is here with me. Hi, Rob.
Hi, Andeka. Thanks, and it's a pleasure to co-present this webinar with you today. A quick introduction for myself. So my name is Rob Martindale, and I'm the EMEA Product Sales Manager here at Masterflex. I have over 20 years experience in manufacturing within the medical device industry. And Andeka, I'm really looking forward to your presentation that you've prepared for us today. And towards the end of your presentation, I'll join in with some updates on our latest pump innovation, the MasterSense LS peristaltic pump. But in the meantime, over to you, Andeka.
Nice. Thank you, Rob. So hello again to all of you. I will introduce myself a little bit more before we start. I am Andeka Ocio, and I studied biotechnology. I have spent the last 2 years working at Progenika Biopharma, a Grifols company, as a manufacturing and process validation technician. Grifols is one of the worldwide leaders in EMEA-based products. Progenika is specialized in developing molecular biology tests. In Progenika, during the last 2 years, I have implemented in-process quality controls, scale up different processes, help to minimize manufacturing errors and increase process efficiency, also reducing the cost of the processes. Therefore, I would like to share my recent experience so that people can learn from it and hopefully do it much better than me. So before getting into the heart of the matter, I have 2 quotes here in this slide, which I really like. Sometimes we tend to leave what surrounds us unaltered because we are afraid of screwing up what we already have, what we obtained in the past. And here goes the first quote: there is nothing permanent but change. Change is positive because change is the possibility to improve, and change is also inevitable. It's not my goal to get philosophical. It's just that technology goes ultra fast and we must adapt to it. And the second quote says, the most dangerous phrase in a language is, we have always done it this way. It is linked with the previous one, with the previous quote, and it is something I have heard many, many times, and that unfortunately happens too often in the industry. I know that it is more comfortable to stick with a methodology forever because changing demands a lot of effort, but you cannot stick to a process just because you have always done it that way. If you have done something always in the same way, it means that you have not improved at all. Luckily, we are not that type of person, and we are open to try new things and learn how to improve the process. This picture on the right shows the main lab where I work in the production area of Progenika. My job there is to improve and validate new and existing bioprocesses. And while shortly, a process is a sequence of independent but linked procedures which consume some resources to convert inputs into outputs. We will be talking in the next minutes about the filling process of the final reagents that go to our clients in Progenika. So the input in this case would be our bulk reagents, and the output, the final containers in which those bulks will be dispensed. The key aspect to analyze today is how that process can be done, which are the different options, and a specific example will help me explain how a process could be improved and some guidelines to do it. So let's start. This is how I improve together with my colleagues in Progenika, a key production step in our factory. The story of how we switched from filling tubes with manual pipettes to using peristaltic pumps in order to take the filling process to a new level. Process improvement is tightly related with process validation. You cannot talk about process improvement without mentioning process validation. In fact, process improvement is considered a part of process validation, a step inside it. Process validation is, summarized, the collection and evaluation of data to establish scientific evidence that a process is capable of delivering quality products. As it can be seen on the graph on the slide, the life cycle of process validation, there are three main steps, which are the design, the qualification, and the continuous verification of the process being validated. And part of the third stage, the continuous process verification, would be to monitor the process and evaluate it for potential improvements. You can see there circled in green. I do not want to go too deep in process validation because we have limited time and it's not the main topic today, but I just want to make clear that with a proper process validation methodology, it is much easier to achieve efficient processes. From now on, I will center in process improvement, but it is slightly related with the life cycle that you are seeing here. And much of the feedback that you need to improve the process is coming from the different steps of process validation. I will divide my explanation of process improvement in these three main points shown in the slide, which are probably the main aspects to fulfill the whole process of understanding and solution building that are needed to optimize a process. Those points or guidelines are analyzing your current process, focusing on fixing important risks and mistakes the current process may have, and finally, seek for efficiency. The solution that will be applied to improve the process should always guarantee the maximum process efficiency. Those 3 points are all important, and none of them should be omitted. In the following slides, each of those points will be covered in order to understand what they mean and how to work on each of them. In order to improve a process, the first thing that should be done is to know exactly how your process works, which are its strengths and weaknesses, all its characteristics. You can divide the process understanding step in 2 columns of strengths and weaknesses, as shown in the picture, or just go straight to describing all its characteristics in a list and without prejudging if they are good or bad. I will explain this better with our example of the filling process on the next slide. Just to mention that the information we need for this analysis usually comes from the design step of the process validation. We will see now an example of how to analyze a process, but there is first something I need to mention. I started my current job 2 years ago, but my factory has much more than 2 years, and there were, of course, some improvements before I joined them. We will now see one of those process improvements related to the filling process. When the factory started producing its first lots some years ago, lot sizes back then were very, very small, and manual pipettes were used to do that filling step. We will analyze the filling process with manual pipettes in order to see how it was done and why it needed to be improved. As I said before, the first thing to do is describing the characteristics of your process in order to have a complete understanding of it. In my opinion, the main characteristics of a filling process would be the ones shown here. First, the main equipment used to dispense. In this case, we have manual pipettes that use pipette tips as a consumable. This type of equipment is really precise, over 99% generally, but on contrary, they are quite slow, with an approximate filling speed of 10 seconds per sample. The speed is disadvantaged by the methodology itself, because with these pipettes, you have to take a tip, moisten the tip with a reagent pool a couple of times, take the liquid and dispense it eventually. All that for each tube that needs to be filled in order to guarantee a proper dispensation and good precision. Besides, the volume range of these pipettes is very limited. Even the larger ones do not usually take more than 10 milliliters. The ones we had were able to dispense from 0.2 microliters to 5 milliliters. So we could now sort out those characteristics between strengths and weaknesses, being their high precision, their best asset, and the low volume range and speed, the main defects. You can obviously perform this analysis with much more detail and more characteristics, such as sterility of the process or complexity of the equipment. It will just depend on the depth you want to give to the analysis, but we will stick to the basics. Nevertheless, a key factor that must be taken into account is that there are not just good and bad processes. There are processes that fit your demands and processes that do not fit them. Therefore, we also have another equally important type of characteristics, the demands of the process. Although there are more, you can see there on the right the three main demands of our filling process: lot size, target volume, and precision. Starting with the lot size, as I said before, lots were very small in our factory in its beginnings, between 25 and 50 containers. Then the target volume was from 1 to 5 milliliter, and the precision needed was above 95%. The speed is also an important demand, but with tiny lot sizes, it comes to a second place, as it does not affect too much. So to sum up, using manual pipettes did fit our demands for some time, when lot sizes were very small and the reagents produced were all lower than 5 milliliters, but the process was not prepared for the future, and that is a mistake. It is essential to be able to easily scale up a process or adapt it to newer demands. Manual pipettes have a clear limitation to scale up, and that was the volume range and the speed. It did not take long to decide that the filling process needed a change. We started to receive an increasing number of orders to buy our products. New products were also developed, and we were asked for bigger containers and kit sizes. The one on the image is one of our diagnostic kits that we sell. As it can be observed, there are different container sizes. We have there 100 milliliter bottles, those big ones on the back, also a 16 milliliter bottle, the green one on the front, then 10 milliliter bottles on each side, the white and black ones, and finally many 1 milliliter tubes on the right. So there are many containers per kit and of different sizes. And manual pipettes were not an option anymore to dispense this type of containers. And well, which was the solution applied in my company? Changing the filling equipment directly. So I will talk now about the filling equipment that were being used when I started working in Progenika. We have two different ones here. On one side, dispensing pipettes that use tips as a consumable, but in this case, a larger volume can be taken in each tip, and then that volume is distributed in multiple dispensations, so that with one aspiration, multiple dispensations can be done. And then the other equipment, the black one on the right, is a 100 milliliter dispenser that is directly attached to a bulk container, and pulling the tip up and pushing down liquid is pumped to the final container. The precision of this equipment is also close to 99%, varying a little bit depending on the target volume. Then the speed of the dispenser pipette is really fast, but the 100 milliliter dispenser is not at all. These two equipment together allow to have a much wider volume range from 1 microliter to 100 milliliters, which is a good strength if considered together. As regards the individual strengths and weaknesses, they are both very precise. The pipette is very fast but needs to be refilled, and the 100 milliliter dispenser is very slow but does not need to be refilled. So why did I analyze these filling devices after the manual pipettes? Because I wanted to show how the filling equipment we just analyzed improved so much the filling process in my company, but still did not take into account some risks and mistakes that both manual pipettes and this dispenser that I have just explained all have. We will keep on with the next key parameter to take into account when improving a bioprocess. We have done the analysis of it, both the equipment and the process demands. So let's go now with the risks and mistakes. I mentioned before that process validation is tightly linked with process improvement. In the first stage of process validation, the process design potential risks are identified, and an FMEA is usually performed. We can use that FMEA to learn which aspects of the filling process need to be improved. FMEA means failure mode effect analysis, and certainly in an FMEA, what you do is to think about potential risks your process may have. You think how those risks could happen, in which different ways, and how those risks could affect the process. Then the risks are studied in order to mitigate them. There are different ways of doing an FMEA, but the one I currently do looks like this. In order to evaluate the risks, different numbers are given to different aspects of the risk. One way of doing it is evaluating them by their severity, letter S, which is the harm that that risk may provoke, and probability, letter P, how often that risk may happen. Then they are multiplied, and depending on the value the result gives, the risk is more urgent to be fixed, or it may be even optional to mitigate it. Currently, it is recommended also to add another variable called detectability, letter D, which indicates the capacity of detecting that error because it goes to the client and how fast it is detected. Well, the thing is that in a process there are a lot of potential risks, and a filling process is not an exception. However, it is difficult to foresee all of them. Normally, all the possible ones are included in the FMEA, and the ones detected during manufacturing are later on included and mitigated. Sometimes risks are also underestimated, and that has happened to me before. I'll give you an example now. So we can assume that in all filling processes, there is a chance of having a final container empty because the operator has forgotten to fill it, or the equipment maybe we are using did not work properly. However, in our case, I assume that the probability of happening that was very low, as the containers go into a kit manually after being filled. So I assume that if the operator takes an empty tube, that it would be clear that it was empty, and he or she would not introduce it inside the kit. All humans make mistakes, right? So the operator and me were just an example of it, and an empty tube reached the final kit and was sent to the client with the corresponding complaint, of course. So we set a mitigation for it. In order to solve the risk of leaving an empty tube or bottle, we implemented a weight control. Weight control is a cheap and efficient process control when our variable to control can be measured by weight. I will briefly explain this. There are usually two ways to perform this weight control. When the final container that we dispense is part of a kit of reagents, we could apply this weight control to the whole kit. That is something we used to do. So the methodology could be to weigh some well-dispensed kits to set the approximate target weight of the whole kit, and then weigh all the kits. If the weights differ more than a certain amount, this depends on the volume of the bottles and the dispensation error that we want to detect. So if they differ more than a certain amount, we can assume that there is an empty bottle. It is a very cheap process control which can eliminate the possibility of having an empty final container or a bottle with less volume than the desired one. In our case, the container with less volume in the kit is of just 1 milliliter, as I showed before. So if we set the limit of the weight control in 1 milliliter in order to detect an empty tube, it is easy that the sum of small little volume variations on the containers can end up being over 1 milliliter. So this method, you may end up opening a lot of kits to see if there is an empty tube by this weight control and find that they are all okay. But that is better than having the risk of sending an empty tube. Another choice could be weighting the kits without the 1 milliliter tubes and weighting the 1 milliliter tubes apart in groups of 20 or 30. So it could be easier to detect those 1 milliliter variations. This could work better than the previous method, but it also takes more time. Then another important risk that is usually forgotten and many filling processes end causing is the repetitive strain injury. As its name says, it is an injury suffered after repetitive movements and overuse. Manual pipettes and manual dispensers require the operator to push a mechanism in order to dispense, and repeating that movement with the thumb or the whole hand hundreds and hundreds of times each day ends causing a repetitive strain injury. We did not realize of this problem in my factory until a worker had to ask for the sick leave because she could not work with her hand as it was injured. And a mitigation for this problem was temporarily to rotate the operators that dispense in order to minimize negative effects. However, as lot sizes were increasing due to growing demand, another solution was needed. So, we have said until now that to improve a process, it is important to understand it completely, all its characteristics. Also, to try to mitigate all possible risks and learn from made mistakes. And finally, it is important to look for efficiency. Sometimes improving different aspects of our dispensing process is enough, mitigating some risks, adding in process controls, hiring more workers, or asking the most efficient workers to teach their tricks and advices to the rest of the workers. All that is fine and can be enough for your current process, but before taking actions, it is essential to evaluate if those measures will be worth or not. What I mean with efficiency is to plan carefully all possible measures to save as much time, money, and resources as possible. Or, in other words, to obtain a good quality product in the shortest time possible and at least cost. Okay, there we go. How did we improve the filling process in our factory? This is the process improvement that I personally did. We moved into a completely new technology for us, the use of peristaltic pumps, specifically a Masterflex LS600. We already had some limitations in our processes with the dispensing pipettes and the 100 milliliter dispensers. Lot sizes kept growing until 1,800 in some products, so we needed the faster equipment which could also adapt to the target volumes and the precision demands. And we also wanted to mitigate in a more efficient way some of the risks filling processes have, such as the ones I already mentioned, leaving a tube empty or getting the operators injured by the continuous use of the filling equipment, all in order to achieve a more efficient process. So I will explain now which major aspects did the peristaltic pump improve and some evidence of it. Okay, let's go with the speed and precision of this peristaltic pump. In our pump system, the speed can be adjusted depending on the needs, from 0.25 to 3,400 milliliters per minute. So the target volume in which we needed the most significant speed change was in the 100 milliliter bottles. With the dispensers, it took 10 seconds for each of them. With the peristaltic pump, it just takes around 4 seconds for each bottle. So we could do it even faster, but as the reagent we dispense creates a lot of foam, we had to limit the flow rate to avoid that foam. As it can be observed in the control chart on the image, good precision was maintained. It has just a small variability and widely meets our demand of a precision over 95%. The smallest tubes take less than a second to be filled, and good precision is also maintained. And then another good point of the peristaltic pump is that volume range is actually unlimited. It may vary in precision and finish time a little bit, but it can be dispensed at almost any volume. But what I like the most about this LS600 pump when incorporating into our process is its ability to adapt to completely different scenarios. It has two different pump heads, one focused on high precision and the other on high performance, although both of them obtain good precision and speed values. The main difference of the pump heads is that each of them works with a different range of tubing sizes. We have been talking all the time about the filling process, but I wanted to mention also an alternative use for these pumps that we found out. So as you see in the performance head and the biggest size tube, the pump goes up to 3.4 liters per minute. We are using it now in my lab to transfer big amounts of liquid. In the slide, it is shown an assembly that we have made in my factory. There on the right, it is a metal structure with 3 floors. In the first 2 floors, bulk reagents are located in their original containers, as we receive them from the supplier actually. And above them, we have our peristaltic pump on the left and an intermediate big container on its right, which is currently of 200 liters size, but it could be easily scaled up. So liquid could be directly dispensed from that intermediate container of 200 liters later on into some final tubes or bottles. Okay, going back into our filling process with the peristaltic pump. The functionality of the pump itself works as a mitigation for some major risks we found out with other equipment. First, the repetitive strain injury I mentioned before. It is completely mitigated with the pump, as the operators do not need to do any effort while dispensing. They just hold the tube and it dispenses. And the other risk I mentioned before was leaving a tube empty, which was being mitigated with a weight control, but it took a lot of time. So we were looking for an alternative, a more efficient mitigation. I'll mention that the pump has different operational modes, time dispense, volume dispense, and continuous mode. But I focus on the volume dispense mode, as it was the natural solution to mitigate our risk. In this operational mode, you select the volume you want to dispense, and you can also adjust some parameters such as the batch amount, which is the key to mitigate empty tubes. If the lot size is 1,000 vials, you just need to set those 1,000 in the pump through the display, as it is indicated in the photo there in the red circle. When the operator starts to dispense, the counter will move up its dispensation, and therefore the operator will always know how many containers has filled. If after the pump finishes dispensing those 1,000, there is an empty vial, it means that the operator has dispensed it twice into one of them. And also the other way, if the pump says we still have some dispensations to make and we do not have more tubes to fill, it means we have skipped some of the tubes, and therefore there are some empty tubes. So we can solve the problem at the moment, and we do not need a weight control anymore, saving a lot of time. So that would be my part.
Andeka. Thanks, Andeka. It's really, really interesting to understand the details involved within process validation and the decision-making process that you, and I'm sure other people in similar positions, go through, looking at the accuracy, risk mitigation, and process efficiency in order to really get the best results from your process. And for me personally, I'm very happy that your journey and decision-making process has led you to choose the Masterflex peristaltic pump range. So now I'd like to take this opportunity to take a quick look at our latest pump. And Andeka, if you would be so kind, as I run through the new pump features, would you be happy to give us all some feedback or commentary on the new features available with the MasterSense LS pump from a user's point of view?
Yeah, sure. No problems about that. Let's go with it.
Excellent. Okay. So the latest innovation within the Masterflex product portfolio is the MasterSense LS peristaltic pump. And a great way to summarize the new MasterSense pump is easy and accurate, with our development team going to great lengths in order to deliver a pump that is both easy to use and highly accurate. So let's run through some of the standout features within the pump. So you can see on the screen the new 5-inch color touchscreen with software that is so intuitive to use. It really is a delight to operate. And I can tell you, Andeka, that the touchscreen is also glove or clinical glove compatible, and I know from your process you use gloves, so and Andeka, nothing to worry about there.
Okay,
any thoughts on that, Andeka? Touchscreen control?
Yeah, touchscreens are cool, so it is visually appealing, and I'm sure it will be user friendly as well. So if it is compatible with gloves, that's perfect for my part.
Brilliant, good to hear. And one feature I know you'd be interested in is the save and recall up to 50 commonly used protocols. And Andeka, I know you're using volume dispense at the moment. Any thoughts on this new feature?
Yeah, I really like that. I mean, it will help to reduce manufacturing time. Currently, operators need to introduce all the pump parameters manually based on the protocol we give them. So saving protocols in the pump itself could allow them to just click on the protocol on the touchscreen and run the program. So it would reduce really the times. And also, even more important, it could also eliminate the risk of setting up wrong parameters. So that's quite useful, yes.
Perfect. So reducing potential human error. Yep, great. That sounds good. And one feature I'd like to just drop in also is the anti-drip feature, which is available on our current LS range of pumps, and that just prevents any media or fluid dropping out of the peristaltic tube at the end of a batch. So that's another feature there. Okay. Quickly going through these features here. So over the air firmware updates. So just like your smartphone, you can receive updates without any manual intervention, quick and easy, and future-proofing the buyer's investment within the pump, similar to a smartphone. 3-level security, so you can set user, super user, and admin security levels. Andeka, anything to add on that? Is that a feature you'd be harnessing?
Yeah, I think it is very useful and necessary at the same time to have security levels if you are implementing the program saving we just commented. So thinking about my own situation, I guess I could load the programs in the pump as an admin, so using the admin user level, and then just give user like the standard user level to the operators so that they cannot change the programs accidentally, right? So yeah, that
Sure.
sounds cool.
Brilliant. That's great to hear. And then another feature would be calibration strength. This is a great feature that gives the user live feedback on the strength of the pump calibration. So a good analogy to explain this feature would be the software used within a password. So if you don't use enough letters, numbers, or symbols when setting up a new password, the software would advise you that the password is weak. So this is the pump does the same function here. If the calibration is outside of our advised tolerances, if you're not dispensing enough volume within the calibration period, the pump would advise you that the calibration is weak, strong, or the best possible sort of parameters. And then just to finish off on the features, so the new MasterSense pump comes with the Masterflex Live software, Ethernet IP, and Profibus. And Andeka, I know you're moving towards using the Masterflex Live software control. Andeka, why are you choosing to go towards the Masterflex Live software?
Yeah, that's correct. We are currently moving towards this Masterflex Live. Especially due to the data recording option that the Masterflex Live web software allows. So it does record which protocols, which ones you are using for how much time and which operator. So I really like that. We're moving towards it.
Excellent, brilliant, Andeka. Thanks very much for the commentary on the new features of the pump. So yeah, keep your eyes peeled, and it will soon be launching.
Glad to hear that. Yeah.
Yeah. And I'm aware we're running short on time, so I'd just like to end on this slide, which is the contact page. So if you'd like any more information on the Masterflex product portfolio, these are the contact details, and of course you can follow us on LinkedIn.
Great, thanks. So before we get to some audience questions, we do have some questions for the audience. So on your screen, before the Q&A starts, we'd like you to go ahead and select your answers for the following questions. Join in and analyze your process. What is the main piece of equipment or technology used to dispense media in your process? Pipettes or manual dispensers, peristaltic pump system, syringe pump system, or other. And then, could a peristaltic pump with protocols such as volume dispense, time dispense, and anti-drip enhance your process? Yes or no? And are you in the process of increasing the yield within your application and would like more information on how the Masterflex product range could help with these requirements? Yes, no, not sure. So again, if you could click and answer the questions on your screen. You may need to scroll down to get to the third question. That would be great. Okay, so we do have some questions from the audience. The first question is, the consumables of the pump are just tubes. Can they be reused or are they single use?
Okay, I think I can answer that. So thank you for your question first. And yes, the tubes are the only consumable that you need to run the pump. Then, can they be reused? Well, from my experience, you can't reuse the tubes, of course. They are quite resistant to hours of work. In my lab, when we started using the pump, validating the process and testing everything, we tested the tubes in order to see if they could be reused several times. So we ran a tube for many hours in order to see if the performance went down, but it resisted quite well, maintaining good precision and also the performance. So anyway, if you want to reuse the tubes, please, you need to clean them. So in our case, we validated the cleaning methodology for the tubes. Also, we limited the use of the tube to one and only reagent. So after dispensing a reagent, we clean the tube and the tube is ready for another run. But we only use that tube for the same reagent in order to eliminate the risk of contamination. Then as regards the validation of the cleaning process, it can be as easy as cleaning a tube and then running water through it. So you can check conductivity of that water in order to detect possible contamination. It is an easy process, but also cleaning the tubes requires some time. And we found out that for big product lots or expensive reagents, it is more worth to throw away the tube and save that time you need to clean it than the money itself that you save. So it is something to value. Well, so to sum up, can they be reused? Yes, definitely. You can clean them and use them many times. Then, is it worth? Well, it will depend on your own process characteristics. Thank you for your question.
Thanks, Andeka. So the next question is, how long can the consumable tube run for?
Great, I can answer that one, I think. So depending on the duty of the pump, for example, a pump running for 2 hours per day, the tube could last for several months. And we generally find that it's the process quality control that governs the rate the tube is changed, as opposed to the necessity to change the tube through tube wear and tear. Great question. Thank you.
Okay, and Andeka, when you went through a decision-making process where you decided that using pipettes was no longer viable and that moving to a peristaltic pump was more efficient and cost-effective, what advice could you give to somebody who is currently looking at their process to become more efficient and potentially save money?
Okay, thank you for the question. In my case, as I explained before, the decision was forced mainly when lot sizes became too large for the all dispensing equipment, and also to reduce the risk of having empty bottles reaching our customers. So also to eliminate the weight control when we had it implemented as well. So because it took a lot of time. We also looked to reduce dispensing time, and finally we ended up moving to a pump option. So I could suggest others to perform the analysis steps that I explained in this webinar, to analyze their process in order to detect which aspects need to improve, focusing also on reducing risk and taking into account process efficiency. And then it is useful to make some research on the market to find the best option that fits your process. Thank you for your question.
Okay, so it looks like we have time for just one more question. If we didn't get to your question, we will be passing your questions on to our speakers. So go ahead and continue to type in your questions. But the last question for this webcast is, how much time does it take to set up the pump prior to dispensing?
I can answer that if you don't mind, Rob. I'll say that from my experience, around 15 minutes more or less. In order to start dispensing, you need to add the tubing, set the parameters, calibrate the pump, and just start running. So I could say that around 15 minutes is a good estimation, maybe less. It will highly depend on how fast you calibrate the pump in order to fit your demands. What do you think, Rob?
Yeah, no, I'd agree. I mean, about your experience there, Andeka. But I could add to that and say that with the release of the new MasterSense pump, with the save and recall feature, where you can recall up to 50 commonly used protocols, that I would imagine just recalling the new protocol or the commonly used protocol would save the user some time. And Andeka, I know we're working on getting you the new MasterSense pump, and I'm sure you would find the benefits when it finally arrives.
Yeah, definitely. I didn't remember that, but yeah, also saving the programs will save a lot of time setting up the pump. So really eager to try that MasterSense. Thank you.
Yeah, brilliant. Thank you.
Well, thank you both, and thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.